A space agency studies distant planets, not seafloor mud sitting on its own doorstep. NASA had no submarine, no funding tied to ocean research, and no obvious reason to show up. Yet when a filmmaker became the first person to solo dive to the deepest point on Earth, a NASA scientist was standing there waiting, ready to study exactly what came back up. What he was actually testing for still says something unsettling about how common life's ingredients might be. The question NASA actually got asked. In March 2012, filmmaker James Cameron piloted a specially built submarine called the Deep Sea Challenger down to the Challenger Deep, the deepest point in the Mariana Trench, becoming the first person to complete that specific dive alone. Waiting to greet him when he surfaced was Kevin Hand, a NASA astrobiologist working at the agency's Jet Propulsion Laboratory, who had joined the expedition specifically to study whatever Cameron managed to bring back up from the seafloor. Hand had already worked with Cameron once before, diving together during an earlier IMAX project called Aliens of the Deep, and Cameron personally invited him to join this new expedition and analyze the water and sediment samples collected during the historic dive. That invitation reflected something genuinely unusual about how a space agency approaches ocean research, since Hand's actual job title at JPL was Deputy Chief Scientist for Solar System Exploration, a role focused on planets and moons rather than anything happening on Earth's own seafloor. Hand's plan involved something that sounds almost backward at first. He intended to subject the samples pulled from Earth's deepest ocean trench to the same kinds of temperatures and radiation levels known to exist far away on the icy surface of Jupiter's moon Europa. Understanding why a NASA scientist would treat samples from the bottom of the Pacific Ocean as a stand-in for conditions on a moon hundreds of millions of miles away means understanding exactly what NASA had already discovered about Europa years earlier. Why a space agency cares about the ocean floor at all? NASA's Galileo spacecraft first confirmed in 2003 that Europa, one of Jupiter's largest moons, hides a genuine liquid water ocean beneath its icy outer shell, and ocean researchers now estimate holds roughly twice the total volume of every ocean on Earth combined. That discovery turned Europa into one of the most seriously considered candidates anywhere in the solar system for supporting life beyond our own planet, since liquid water ranks among the most basic requirements scientists look for when assessing whether any environment could plausibly host living organisms. Studying an ocean sealed beneath miles of solid ice on a moon, this distance presents an obvious problem, since no current spacecraft can simply drill through that shell and swim around collecting samples the way a submarine can on Earth. Researchers instead rely on analog environments, places on our own planet that share enough physical similarities with these distant alien oceans that studying them can reasonably predict what conditions on Europa or similarly icy moons might actually be like. Earth's own deep ocean trenches turn out to match several of Europa's key conditions surprisingly well, sharing extreme cold, crushing pressure, and complete darkness with no sunlight reaching anywhere near the bottom. That overlap is exactly why a NASA astrobiologist would show up at the surface waiting for a filmmaker's submarine, since the samples Cameron pulled up from 7 miles beneath the Pacific offered Hand a genuine physical stand-in for material that might one day get collected from an ocean nobody has ever actually reached. The dive that put a NASA scientist on the team. Cameron's submarine, the Deep Sea Challenger, descended to the Challenger Deep in 2 hours and 36 minutes, then spent several hours exploring terrain nobody had ever directly observed before making a roughly 70-minute return trip back to the surface. That entire round trip demanded engineering built specifically to survive pressure that would instantly crush an ordinary submarine hull, since the same brutal conditions crushing steel at that depth apply equally whether the vessel belongs to a research institution or a solo filmmaker funding his own expedition. Once Cameron's samples reached the surface, Hand's analysis focused specifically on testing how the water and sediment collected from Earth's most extreme ocean environment held up when exposed to conditions matching what instruments have already measured on Europa's own icy surface. That kind of direct, physical comparison between a genuine terrestrial sample and known extraterrestrial conditions represents exactly the kind of concrete, testable science astrobiology depends on, rather than pure speculation about what might theoretically exist somewhere else in the solar system. This entire expedition demonstrates something worth sitting with directly. A NASA scientist did not travel to the bottom of the ocean to search for actual living organisms swimming around down there, expecting to find something dramatically new. He went to study the physical and chemical environment itself, treating Earth's own deepest trench as a working laboratory for understanding an ocean nobody has any current way to visit directly. Understanding why that specific approach made sense, scientifically, requires looking at a much older discovery that first convinced NASA these deep-sea environments were worth taking seriously at all. The discovery that changed how NASA thinks about life. Until 1977, most scientists believed every living thing on Earth ultimately depended on sunlight for energy, since plants use sunlight directly and nearly every other organism eventually gets its energy by eating something that ate a plant somewhere further up the chain. That assumption collapsed the moment researchers piloting the deep-sea submersible Alvin, operated by the Woods Hole Oceanographic Institution, descended along the Galapagos Rift and discovered hydrothermal vents scattered across the ocean floor, finding thriving communities of large, strange organisms clustered directly around plumes of mineral-rich, superheated water erupting from cracks in the seafloor. The scientists aboard that expedition had actually gone looking for something else entirely, expecting to study underwater volcanic activity rather than biology. What they found instead genuinely stunned the research team. Dense colonies of foot-long tube worms, giant clams, and other organisms nobody had ever seen before, clustered tightly around vents in water reaching temperatures far above anything researchers expected life could tolerate anywhere near that kind of geological activity. The specific tube worm species eventually identified from these vent communities, later named Riftia pachyptila, turned out to have no mouth and no digestive system at all, surviving entirely through a symbiotic relationship with internal bacteria that convert vent chemicals directly into usable energy on the worm's behalf. These vent communities survive through a process called chemosynthesis, where specialized bacteria convert chemicals dissolved in the vent water directly into usable energy, completely independent of sunlight, supporting larger organisms like tube worms and clams that feed on those bacteria in turn. Some of the microorganisms living at these extreme depths, called barophiles, have developed specialized waxy protective layers specifically to survive the crushing pressure found at these depths without their cells collapsing. That single discovery mattered enormously for astrobiology specifically, since it proved for the first time that a genuine functioning ecosystem could exist without any sunlight reaching it at all, relying purely on chemical energy instead. Scientists studying the possibility of life on distant, ice-covered ocean moons immediately recognized the implications, since those same moons receive essentially no useful sunlight beneath their thick ice shells either, making Earth's own chemosynthesis-dependent hydrothermal vent communities the single closest working example available anywhere of exactly the kind of life that could theoretically survive on a completely different world. The moon that made this search urgent. Europa's confirmed subsurface ocean turned this theoretical possibility into a genuine, funded scientific priority, eventually producing NASA's Europa Clipper mission, the largest spacecraft the agency has ever built, specifically for a planetary science mission. Europa Clipper launched on October 14, 2024, lifting off aboard a SpaceX Falcon Heavy rocket from Kennedy Space Center in Florida, beginning a journey covering roughly 1.8 billion miles before the spacecraft finally reaches Jupiter in April 2030. The spacecraft carries 9 separate science instruments, plus a gravity science experiment built directly into its telecommunications system, covering everything from ultraviolet and thermal imaging to ice-penetrating radar and mass spectrometers designed to directly analyze any material the spacecraft encounters near Europa. Rather than orbiting Europa itself, which would expose the spacecraft to dangerously intense radiation surrounding Jupiter for extended stretches of time, Europa Clipper will instead orbit Jupiter directly, periodically diving through that radiation belt to complete 49 separate close flybys of Europa, some passing as near as 25 kilometers above the Moon's icy surface, before pulling back away from the radiation again after each individual pass. That specific mission design, repeatedly diving close and retreating rather than settling into a long-term orbit, lets Europa Clipper study its target across several years, rather than being destroyed by radiation damage within days or months. The mission's stated primary science goal is stated in refreshingly plain language directly on NASA's own website to determine whether there are places below the surface of Europa that could actually support life. Precisely the same underlying question Kevin Hand had already begun investigating using Challenger Deep samples more than a decade before Europa Clipper ever left the launch pad. If you're enjoying this deep dive, hitting subscribe means you won't miss the next one. Europa is far from the only icy moon drawing this same kind of serious astrobiological attention, though. A separate moon, considerably smaller and orbiting a completely different planet, has already provided scientists with something Europa never has: direct physical samples of its own hidden ocean delivered straight into the path of a passing spacecraft. The icy moon that sprays its own ocean into space. Saturn's small moon. Enceladus turned out to hold one of the most direct, physically sampled pieces of evidence for a genuinely habitable alien ocean found anywhere in the solar system. NASA's Cassini spacecraft discovered dramatic geyser-like jets erupting from cracks near Enceladus's south pole, later confirmed to be spraying material directly from a global, salty, liquid ocean hidden beneath the moon's icy surface, shooting particles into space at roughly 800 miles per hour. For decades before Cassini's arrival, scientists had puzzled over why Enceladus reflected sunlight so brightly compared to every other body in the solar system, and why a faint ring of ice particles circling Saturn, known as the E-ring, seemed to trace directly back toward this one specific small moon. Cassini finally solved both mysteries at once, confirming that fresh ice constantly resurfacing Enceladus and feeding that distant ring both originated from the exact same source, vents connected directly to the moon's hidden global ocean underneath. Some of the ejected material falls back down onto Enceladus's own surface, while the rest escapes entirely to help build that same distant ring circling Saturn. Cassini's instruments flew directly through these plumes multiple times, detecting water, ice, simple organic chemicals, and critically, molecular hydrogen, a specific chemical signature that on Earth typically forms when hot water reacts with iron-bearing minerals deep inside active hydrothermal vent systems. Cassini also detected tiny silica nanograins passing through the plume material, a specific type of particle that can only form when liquid water and rock interact at temperatures above roughly 90 degrees Celsius, providing direct physical evidence that genuinely hot, active hydrothermal chemistry is happening somewhere on Enceladus's hidden seafloor right now. Researchers have since detected or tentatively detected all 6 of the basic chemical elements considered essential for building the biological molecules life depends on: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur, present somewhere within these plume samples. A team of researchers published a formal statistical analysis in 2021 specifically testing whether the observed rates of hydrogen and methane escaping from Enceladus's plumes could be fully explained by ordinary rock chemistry alone, without any biological process involved. Their Bayesian statistical model found that pure rock chemistry could not fully account for the observed escape rates on its own, that the results remained fully compatible with conditions habitable for methane-producing microorganisms called methanogens, and that a genuine biological explanation actually scored as the most statistically likely scenario, assuming the underlying probability of life having emerged there in the first place was reasonably high to begin with. What that actually means for the unsettling answer. None of this research has produced confirmed proof that life actually exists on Europa or Enceladus. Scientists studying both moons have been consistently careful to describe their findings as compatible with habitable conditions rather than direct evidence of living organisms, a distinction serious astrobiology research maintains rigorously rather than overselling every promising chemical signal as confirmed alien life. What this research has genuinely established is considerably more unsettling in its own quiet way than a dramatic monster reveal ever could be. The specific chemical and geological conditions capable of supporting life without any sunlight at all, conditions scientists once assumed were rare or impossible anywhere, turn out to exist naturally, both at the bottom of our own planet's oceans and, based on real physical sample data, apparently within at least one other moon orbiting a completely different planet. If the basic recipe for life-supporting chemistry shows up this readily in at least two separate places researchers have actually managed to sample directly, that raises a genuinely serious statistical question about just how many other ocean worlds scattered across the universe might already be quietly meeting the same basic requirements. That is the actual unsettling answer sitting underneath this entire research effort. Not a specific monster or artifact, but a growing body of real, carefully gathered evidence suggesting the raw ingredients for life may be considerably less rare across the universe than humanity assumed for most of scientific history. Getting a full, honest picture of that question back here on Earth first requires understanding exactly how little of our own ocean floor scientists have actually managed to see with their own eyes. How little of our own ocean floor. We've actually seen a popular claim repeated constantly in documentaries states that humanity understands the surface of Mars better than the bottom of its own oceans. In May 2025, oceanographer Catherine Bell and her research colleagues published a study in the journal Science Advances, specifically calculating exactly how true that claim really is, rather than simply repeating it as an unverified saying. Their results were genuinely striking. Scientists have visually observed less than 0.001% of the entire deep sea floor, an area roughly equivalent to 1/10 the size of the small European country of Belgium, out of an ocean floor covering the majority of the entire planet's surface. That number represents actual visual observation using cameras and human eyes, a considerably stricter standard than broader sonar mapping efforts, which have covered a noticeably larger share of the seafloor, without ever actually showing researchers what that terrain visually looks like up close. That genuinely tiny sliver of direct observation means every single discovery described throughout this entire video, the hydrothermal vents, the strange deep-sea creatures, the pollution already found at extreme depths, comes from studying an almost unimaginably small fraction of the actual ocean floor. Whatever else remains hidden across the roughly 99.999% of the seafloor, nobody has ever actually looked at directly is, by definition, still completely unknown. A genuinely humbling thought once you sit with the actual math behind it. What actually lives down there on Earth? Within that tiny observed sliver, researchers have already documented remarkable, specialized life clearly built to survive extreme pressure and total darkness. In 2014, cameras lowered into the Mariana Trench recorded a pale, soft-bodied fish swimming past at a depth deeper than any living fish had ever been filmed before. Formally described in 2017 as a new species called the Mariana snailfish, its body relying on a natural compound called trimethylamine oxide to keep its protein stable under crushing pressure that would destroy an ordinary fish's cells. Sharing that same depth range is a shrimp-like creature called the supergiant amphipod, growing up to 30 centimeters long compared to the roughly 1 centimeter size of its ordinary shallow water relatives. Alongside xenophyophores, single-celled organisms roughly the size of a mango that hold the record for the largest single cell known anywhere in science. Genetic studies suggest that supergiant amphipods living in trenches thousands of kilometers apart carry nearly identical DNA, hinting at a single connected population spread across huge stretches of the world's deepest waters. None of these animals resemble anything from an old sailor's legend. They are small, strange, and built entirely around surviving pressure and permanent darkness rather than dominating anything through size or aggression. That real biological picture, strange but scientifically explainable creatures rather than mythical monsters, matches almost exactly the same careful, evidence-based approach NASA applies when studying Europa and Enceladus, treating each discovery as a specific, testable data point rather than an excuse for wild speculation. One additional discovery from Earth's own deep ocean, though, reveals something genuinely disturbing that has no equivalent anywhere else humanity has ever managed to explore. The pollution NASA never has to worry about elsewhere. In 2017, a research team led by Alan Jameson at Newcastle University published a study in the journal Nature, Ecology, and Evolution, examining small scavenging creatures pulled from the Mariana and Kermadec trenches, two trench systems sitting roughly 7,000 kilometers apart on opposite sides of the Pacific Ocean. What they found inside the fatty tissue of these creatures were extraordinarily high levels of banned industrial chemicals called polychlorinated biphenyls, at concentrations roughly 50 times higher than crabs studied in one of the most polluted rivers in China. Jameson said publicly that his team had always assumed the deep ocean remained remote and pristine, safe from any meaningful human impact, and that these results proved that assumption could not be further from the truth. Once a pollutant like this sinks far enough into the deepest ocean trenches, there is nowhere left for it to go, meaning it simply accumulates indefinitely in one of the only places on the planet nobody visits often enough to ever clean it up. Explorer Victor Vescovo, who has personally piloted multiple crewed dives to the Challenger Deep, has separately reported spotting plastic bags and food wrappers resting on the trench floor, ordinary human trash sitting quietly at the single deepest point on the entire planet. Nothing comparable exists anywhere in the actual astronomical data collected from Europa or Enceladus. Whatever chemistry Cassini detected spraying out of Enceladus, and whatever conditions Europa Clipper eventually confirms once it reaches its target in 2030, none of it carries any trace of human industrial contamination, since humanity has never sent anything capable of physically polluting either ocean the way our own industrial chemicals have already reached the bottom of our own planet's deepest trenches. What comes after Europa, Clipper finishes looking. Even before Europa Clipper's own mission concludes, NASA researchers have already spent years exploring what an even more direct follow-up mission might eventually look like. Engineers working under a project called Ocean World's Europa Technologies have investigated concepts for an actual Europa lander, a spacecraft that would touch down directly on the moon's icy surface, rather than simply flying past it from a distance, the way Europa Clipper is designed to do. A lander of this kind would face an obvious limitation, since its outer shell would prevent it from physically diving beneath Europa's ice and swimming through the ocean below, the way Cameron's submarine explored the Mariana Trench. Researchers have instead proposed positioning a future lander directly beside one of the open crevasses already observed venting water from beneath Europa's surface, letting the spacecraft sample both the surface ice and any escaping ocean water without needing to drill through miles of solid ice first. Engineers have specifically been developing intelligent landing systems, specialized low-temperature battery technology, and compact avionics packages capable of surviving Europa's brutal combination of extreme cold and intense radiation long enough to complete that kind of mission. Europa Clipper's own findings, once its 49 flybys wrap up sometime after 2030, will likely shape exactly where any future lander mission would eventually need to touch down, continuing the same careful, evidence-building approach that started with Kevin Hand's sample analysis back in 2012 and continues through every single flyby Europa Clipper completes on its multi-year tour of Jupiter's icy moon. Why that comparison is the actual unsettling part. Put all of this together, and the genuinely unsettling conclusion, sitting underneath this entire research effort, has nothing to do with monsters or classified secrets. It involves two separate, equally uncomfortable facts sitting directly next to each other. The specific chemistry capable of supporting life without sunlight, the exact conditions astrobiologists spend entire careers searching for on distant icy moons, already exist naturally right here on Earth, thriving quietly at the bottom of our own oceans for billions of years before anyone even knew to look for it. At the very same time, humanity has managed to contaminate that exact life-supporting environment with banned industrial chemicals before ever fully understanding what lived there in the first place, reaching the deepest, most inaccessible trench on the entire planet with our own pollution decades before most scientists thought to seriously check. Meanwhile, genuinely pristine alien oceans sitting on Europa and Enceladus remain untouched by any human contamination, purely because humanity has not yet developed the technology needed to physically reach them, not because we have shown any particular restraint once given the chance. That combination, life-sustaining chemistry proving more common across the solar system than once assumed, paired with humanity's own demonstrated tendency to contaminate exactly the environments most worth protecting and understanding, is the real answer NASA's ocean research actually points toward. It is a considerably more sobering conclusion than any single dramatic headline about deep-sea monsters could ever deliver, and it is built entirely on real, carefully documented science rather than speculation. Verdict: here is where things actually stand. NASA astrobiologist Kevin Hand personally joined James Cameron's historic 2012 solo dive to the Challenger Deep specifically to analyze the samples brought back, treating Earth's deepest ocean trench as a physical stand-in for conditions on Jupiter's moon Europa, whose subsurface ocean was confirmed by the Galileo spacecraft in 2003. That entire approach traces back to the 1977 discovery of hydrothermal vents along the Galapagos Rift, which proved for the first time that life could survive entirely without sunlight through a chemical process called chemosynthesis, the same basic model researchers now apply directly to Europa and to Saturn's moon Enceladus, and which now drives NASA's Europa Clipper mission, launched in October 2024 and set to arrive at Jupiter in 2030 for 49 close flybys of the icy moon. Cassini spacecraft data collected directly from Enceladus's plumes detected hydrogen, silica nanograins, and other chemical signatures consistent with active hydrothermal vents on that moon's own hidden ocean floor. With a 2021 statistical analysis, finding the observed chemistry actually scored as more likely explained by biological activity than by rock chemistry alone. A 2025 study found humans have visually observed less than 0.001% of Earth's own deep ocean floor, even as a 2017 study confirmed that same barely explored trench system already carries measurable levels of banned industrial pollution 50 times higher than one of the most contaminated rivers on the planet. No monster, no classified secret, and no single dramatic discovery sits at the bottom of this story. What sits there instead is real, carefully gathered evidence that the basic chemistry needed to support life shows up in more places across our own solar system than scientists once assumed, sitting directly alongside proof that humanity has already managed to contaminate the one example of that chemistry we can actually reach, decades before we ever fully understood what we were contaminating in the first place. If this kind of deep ocean mystery is your thing, subscribing helps more of these stories reach you the moment they come out.